My name is Darren Griffin. It is such a fantastic honor to be co-moderating the session with my friend and colleague, Stacy Colaco. Stacy is an inspiring young scientist keen on investigating the Y chromosome microdeletions and beyond male infertility. So very, very, very relevant to this session here. She completed a PhD in biochemistry from the Indian Council of Medical Research, Mumbai, where she joins you from today. And she has developed this fantastic, simple and cost-effective assay to diagnose Y chromosome microdeletions in infertile men. I might talk to you about that a little bit later, Stacy. This uses non-invasive sampling techniques, which is fantastic. Now, she's also developing some machine learning-based algorithms that will predict embryonic outcomes when sperm-bearing Y chromosome microdeletions are used in ART. So it's a great honor to have you here, Stacy. Passing over to you.
Thank you so much, Darren. I, in turn, would like to acknowledge your contribution to this field. Professor Darren Griffin received his PhD from University College London. After several postdoctoral stints, he settled at the University of Kent, where he's been for the last 15 and more years. He's worked under the mentorship of some stalwarts like Professors Joy Delhanty, Christine Harrison, Terry Hassel, Alan Handyside, and Malcolm Ferguson Smith. Professor Griffin, we're delighted to know that you're a world leader in cytogenetics and have performed the first successful PGD. More recently, he's played a significant role in the development of carrier mapping. Besides all this, he's a prolific science communicator, making every effort to make scientific research publicly accessible, and is an enthusiastic proponent for the benefits of interdisciplinary research endeavor. Welcome, Professor Darren.
Thank you very much. And you mentioned two very important people here. I'd just like to spend a moment in the memory of Professor Joy Delhanty, who sadly passed away just in the last few weeks, my PhD mentor. And also a little shout out to my first mentor, Christine Harrison, whose images of the X and Y chromosome, she tells me, were the ones that appeared in our advertising material this time, back from pictures in 1982.
Oh.
So absolutely. So Stacy, I believe you're going to introduce our first speaker.
Yes, now it's time to formally begin our session, and I hope you all are looking forward to that. But before we begin, I'd like to remind the audience to please post your questions using the Q&A section of the conference and not on the chat function. It will be great if you can mention your name, where you're from, and to whom your question is directed. And so with this, we can begin. I'm privileged and honored to welcome our first speaker, Professor Jenny Graves, who is an evolutionary geneticist and exploits our distant relationship to Australian animals to discover how genes and chromosomes evolve. She uses this technique, she uses this unique perspective to explore the origin, function, and fate of human sex genes and chromosomes, and has even predicted the disappearance of the Y chromosome. Professor Graves is going to be joining us in real time during our Q&A session. And for now, we have a pre-recorded video of her talk, which we can now watch.
Well, I'm looking forward to giving this talk on the Y chromosome to what sounds like a really interesting and eclectic audience. So I want to talk about the rise and fall of the human Y chromosome. And I want to start with the origin of the human Y chromosome, which was, of course, a perfectly ordinary autosome. So we start with a proto-Y chromosome, which is the same as a proto-X chromosome, and we end up with a much reduced Y chromosome with hardly any genes on it, which is in danger of disappearing. There it goes. So I don't need to tell you that the big difference between female and male chromosomes is 2 copies of each chromosome in the female, but in the male, one chromosome stands out by being unpaired. This chromosome is called the X chromosome. not because it looks like an X, which it does in humans, but not in other mammals necessarily, but it was an X for unknown. And the Y chromosome is male-specific chromosome, a pathetic little thing, as you see, called the Y simply because the X was calling the X. So the Y chromosome has been engaging a lot of people in a lot of thought because it is very weird indeed. Well, we know a lot about the Y chromosome now. It's been sequenced. It's hard to sequence because there's an awful lot of DNA junk on the Y chromosome. And here's a fairly recent picture of the Y chromosome and all its genes. And you'll notice some very weird things about the Y chromosome. Largely that there's hardly any genes on it. So at the very top is a region called the pseudo-autosomal region, or PAR, and it's got lots of genes in it that have nothing much to do with sex. And at the very bottom, there's another little pseudo-autosomal region with a few genes on it. But what's in the middle is male specific, and it's really what counts. And there are only 17 single copy genes on the Y chromosome. But there's also a number of genes that are present in multiple copies in a really weird structure. So there's these great big loops that have multiple copies of some of these genes on them. And these loops are a palindrome, and they can actually pair, and you can get gene conversion from one to the other. So it is a very unusual chromosome in that there's lots of genes at the very top, which is not really Y chromosome, hardly any genes in the middle, and this peculiar structure of amplified genes. So that's the structure of it. The other thing to note is that I've color-coded now the genes that have got partners on the X chromosome. So almost all the single copy genes are all got partners on the X chromosome, and also some of the multi-copy genes in these big arrays have also got X partners, and our labs happen to have discovered some of these X partners. So it's a very strange and very unusual chromosome. So sex chromosomes are weird. They don't look anything like a pair. But in fact, they do share a tiny little region at the top. That's the pseudo-autosomal region, and there's 16 genes in it. But the rest of the Y chromosome, there are only 27 protein coding genes that are specific to the Y chromosome. There are many copies of some of these, but most of them are inactive. So it's full of pseudogenes and genes that don't work very well. And there's several genes with male-specific functions, for instance, in sex and spermatogenesis. And the principal one of these, of course, is the SRY gene, which is what the gene which triggers sex determination by triggering differentiation of a testis in the embryo. So that's the whole purpose of the Y chromosome, to carry this one gene SRY, without which you don't become male. Well, now the X chromosome looks like a perfectly normal chromosome, and it's not exactly gene rich, it's somewhat gene poor, but there's something like 1,000 genes on it that code for proteins, and they have all sorts of different functions in blood clotting and housekeeping functions and so forth, but it's not really normal. There are too many genes on it that have something to do with sex and reproduction, fertility, gonadogenesis. And there's also far too many genes that are expressed in the brain and mutations in which give you mental retardation. And the really weird thing is that some of these are the same genes. I call them brains and balls genes because they're expressed in the brain and the testis. We've got to say, well, why are sex chromosomes so weird? Is it so they work better, or is it a horrible evolutionary accident? And I'm here to tell you that indeed it is a horrible evolutionary accident. And to understand the Y chromosome, we've really got to understand how it evolved. So over the years, I've drawn up models of the X and the Y chromosome, which I'll share with you. The oldest model is that the X chromosome is just really an ordinary chromosome. It's as sweet as it ever was. And there's been no change since it was an ordinary chromosome. But it also seems to contain too many genes that control other genes. So that's my dominatrix X. And it's, as I said, it's a smart X because it has all these genes that are required for brain function. And it's a sexy X too, because there's so many genes on it, about 5 times too many, that are involved in gonadogenesis or fertility. And there are these genes I mentioned to you that do both. So that's the X chromosome, not normal, although it's a large chromosome with lots of genes on it. But the Y chromosome is a really weird one. And people have been thinking for years, well, the Y, it may be small, but it's a really dominant, macho little thing. But we now know that's only because it has the SRY gene on it. And that's why it has this dominant, its male dominant effect. But there are other models. One is that it's a selfish element, that it grabs genes from other chromosomes that are handy in a male. They give a male an advantage by making more sperm or faster sperm or something like that. Well, that actually is the case in Drosophila, but not in humans. There are only a couple of genes on the Y chromosome that came from autosomes that have an effect on male fertility. So my model is different, and that is the Y chromosome is really a bit of a wimp. It's just a pale shadow of its erstwhile glorious self as an X chromosome. So I want to tell you now that the Y chromosome looks like a degraded X chromosome. And the theory behind that is a very old theory. It's not mine. It's 100 years old, that once the X chromosome and the Y chromosome were just an ordinary pair of autosomes going about their ordinary autosomal business and recombining, that's my little dotted lines there, over the whole chromosome. But then one partner acquires a male determining gene, and that was probably SRY, and that was the kiss of death for that chromosome, because once that happened, other genes nearby that acquired a male advantage accumulated, and to keep that whole package of male-specific genes together, recombination with the X was suppressed. And you probably know that that's very bad for any region of the genome not to be able to recombine. And so what happens then is that there's a lot of deletions and mutations that occur in that region that doesn't recombine, and you get very rapid degradation of the Y chromosome. And it's really only the top bit that hasn't yet been degraded. And that's pretty much what our own X and Y chromosome look like now. But things can go further. You can get more and more degradation until the Y chromosome doesn't recognize the X chromosome at all, and they don't pair at meiosis. In fact, they have a completely different way of segregating at meiosis so that sperm gets either an X or a Y. And if you go further, the Y chromosome could even disappear. And that's what I'll end up talking about. So this theory proposes that the Y chromosome is essentially a degraded X chromosome. And everything we know about the Y chromosome confirms that. One thing that we have known for a long time is that most genes on the Y chromosome have partners on the X chromosome. And these are just 3 genes that we have to have discovered the X partner of, but that actually includes the SRY gene. There's a gene on the X chromosome called SOX3. It's a very old gene. It's there in fish and frogs. And it's an interesting gene because it's important in early embryogenesis, and it may have something to do with mental retardation and spermatogenesis. So all these genes on the X seem to have acquired a male-specific function on the Y chromosome. And interestingly, they're all brains and balls genes. So it looks like brains and balls genes on the X have evolved functions on the Y chromosome. And of course, each one of these is an interesting story, of evolution, and I'll just tell you one of them. But something like 21 out of the 27 genes on the Y chromosome have partners on the X. So that certainly supports the hypothesis. So the evolution of the SRY gene itself, of course, has been of a great deal of interest. And many years ago, we discovered the SOX3 gene on the X. We discovered that it was active in the brain and in germ cells, but not in the developing testis. So it's not a sex-determining gene. So what happened, we suggested, and it turns out to be correct, that this gene actually was redirected to be expressed in the gonad. And what we find is that ectopic expression in the testes, both in humans and in mice, gives you XX males, where SOX3 can act as a sex-determining gene. And we proposed that what happened back in evolutionary time was that a breakage upstream of the business part of the gene, that yellow bit, the HMG box, and then a fusion to a gonad-specific promoter. So that's driving the SOX3 expression into the developing testis, and that gives it its new function in testis determination. So it's a very simple way of making a sex-determining gene, and it emphasizes that all you need in this case is a change in the tissue of the expression to have evolved a new sex-determining gene. Well, I want to talk now about the origin of human sex chromosomes, and this is where I started many, many years ago, looking to where were our sex chromosomes 105 million years in an elephant? Where were they 148 million years ago in a marsupial? Where were they in a monotreme 166 million years ago? And so using this evolutionary approach, we could look at sex chromosomes. And of course, the Y chromosome being a degraded X, we look at the X chromosome. So what we've done is to map human X genes in kangaroos and elephants and chickens and all sorts of things. And we came up with some results that we really did not expect. For a start, we worked on kangaroos. I'm Australian, so this was a natural thing to do. And we took genes from the human X and we cloned them. We got back clones from kangaroos with the orthologous gene. And there's two of them there that map to the X chromosome very closely. So we thought, okay, so it's the same on X chromosome. It's not, because once we started looking at the genes on the short arm of the human X chromosome, we found they're all on chromosome 5. So that was a bit of a surprise, and we backed this up by doing chromosome painting. We painted a kangaroo X DNA onto my students' very own chromosomes, and you can see that only the 2/3 of the bottom bit of the human X paints with a kangaroo X. So the top bit is another block. And so when we look at all these genes, we could see there were 2 blocks of genes, one on the X and one autosomal, in a kangaroo that relate to the human X. So if we paint the human X in kangaroo colors, that's what it looks like. Now, of course, that doesn't tell us whether the ancient X was the kangaroo X or the human X may have split up into 2. So we went to an outgroup, chicken, and we discovered the same blocks of genes were separate in a chicken genome, and they were both autosomal, as we expected, because chickens have a completely different way of doing sex. And we also looked at elephants, and they were interesting because they have exactly the same genes in the same order on the X. It's just the centromere has slipped down a notch or 2. So we have a neocentromere in humans and most therian mammals. So what this showed us was that there was an ancient X chromosome of therian mammals, that's marsupials and placentals. And then there was this autosomal bit that got stuck on about 105 million years ago. So there's 2 blocks that have gone to our X chromosome. And because we know that the Y is a degraded relic of the X, we expected to see blue and green genes there as well. The surprise was there's hardly any blue genes left. There's only 4 genes that were there on the original Y chromosome that are still there on the human X. Most of the human Y derives from this bit of autosome that got added on and seems to be pretty much irrelevant for sex. So that should have given us the idea that the human Y would have disappeared entirely if it hadn't had this addition 105 odd million years ago. Well, we wanted to go back further, so we wanted to look at monitory mammals. These are the egg-laying mammals of Australia, and they're a very ancient offshoot. So 166 million years ago, we shared a common ancestor, and we expected to find the same old X chromosome, probably the marsupial version of it. To our great surprise, when we used the markers from the human X, they went to an autosome. They were located on the chromosome 6. There it is in green. We've used a paint to light it up, and the orange spots are SOX3, and all the other genes are there too. So it's the same chromosome, but it's not a sex chromosome because there are 2 copies in males and 2 copies in females. So that again showed us that the origin of our sex chromosome was an autosome, but it was a whole lot more recent than we had expected. And what it says is our X and Y chromosomes are really rather young. They evolved from an autosome only somewhere between 148 and 166 million years ago. So they're much younger than we thought. Well, how do monotremes do sex? I'll just have to tell you that there are actually 10 sex chromosomes, 5 X chromosomes and 5 Y chromosomes in males. And they don't just pair up and segregate at meiosis, they form a chain. So there's pseudo-autosomal regions at each end. They form a chain and all the X's go to one pole and the Y's go to another pole. There's no SRY, and it looks like the sex-determining gene is the anti-Müllerian hormone, which is interesting because some fish have that as well. So they have a very bizarre way of doing sex. When we mapped the sex chromosomes, we found absolutely no homology to human sex chromosomes. However, oddly enough, there's a lot of homology to bird sex chromosomes, and all those green genes you see on my map there are on the bird Z chromosome. So we had to say, well, really, as far as sex goes, platypus is a furry bird, and it may have a version of an ancestral mammal-like reptile sex determining system. And that again says that our X and Y are much more recently evolved. Well, going even further away, we had a look at birds, and one of my students had a look at emus. Emus are rat-eyed birds, flightless birds, and they're very interesting because they have the same sex chromosomes as a chicken by descent, but they don't look the same. So birds are quite different. It's the male that has 2 big gene-rich Z chromosomes. The females have only 1, and they have this pathetic little W chromosome, which is female specific. But it's got somewhat askew here, but rat-eyed birds actually have a W chromosome, which is almost full size, almost the same size as the Z chromosome, but it's still a sex chromosome because there's a gene on it called DMRT1 that's on the Z chromosome, but not the W chromosome, and it's dosage of that gene that does sex. Snakes are interesting. Their sex chromosomes look much the same as birds, but mapping shows they're completely different. We don't know what the sex chromosome is there, but again, there seems to be some snakes that have a W chromosome that's pretty much the same size as the Z, and others that have a very degraded W chromosome. So putting all this together, we can see that all these higher reptiles have essentially the same genome, same genes pretty much, and is only the arrangement on chromosomes that differs. But we can see that there's 5 patches of that common genome that have become sex determining in one lineage or the other. So for instance, the yellow chromosome became a ZW in snakes and the red became the ZW in birds because it has this gene on it, DMRT1, whose dosage determines sex in birds. And monotremes have a version of that, but they have the AMH gene stuck onto that as well. And our sex chromosomes evolved somewhere between 148 million and 166 million years ago, and then a bit got added onto that as well. So that allows us to date the evolution of SRY and the evolution of our X and Y chromosome. Well, what's happening to the poor Y chromosome? It started off with about 1,000 genes between these two autosomal regions, which are still autosomal in platypuses as well as chickens. If we go back 148 million years ago, we can see that the larger block became the X and Y chromosome with the evolution of SRY. And if we go back 105 million years ago, we can see that the additional bit got globbed on. If we look at the present, there's only 43 genes total left on the Y chromosome out of the 1,000 genes it started off with. And if we look into the future, we can actually see that the whole Y chromosome has already disappeared in some lines of rodents. So there's two kinds of rodents that have actually completely lost their Y chromosome, mole voles and spiny rats. And I'll tell you a little bit more about those. So things look pretty grim for the Y chromosome. So why does the poor old Y chromosome degrade? Isn't evolution supposed to keep all the chromosomes in good order and condition and expunge mutants and select advantageous gene combinations? Well, there are two things about the poor old Y chromosome that lead it to very rapid degradation. The first thing is that there's very high variation. And that's the reason for that is that the poor old Y is always stuck in a testis. It never gets into an ovary. And the testis is a dangerous place to be because there's a lot of cell division going on for making sperm. There's something like 300 odd cell divisions instead of 20 or so in an oocyte. So there's much more likelihood of genetic accidents. And we know now that most de novo dominant mutations in humans come through the father for that reason. But that's not the only problem the Y has. Selection doesn't work very well on the Y chromosome because it doesn't recombine with anything. There's got nothing to recombine with. And that means it can't put two good bits of the Y chromosome together to make a whole good Y chromosome. Once there's a mutation on the Y, it's stuck with it. And so you get this ratchet-like accumulation of mutations as you accidentally lose Y chromosomes, which are less mutated. So there's no repair and no recombination. So that's why Y chromosomes, whether in fruit flies or in humans, degrade extremely fast. So we can calculate when the Y chromosome is expected to disappear because we know about 150 million years ago, there was something like 1,000 genes on it, and now there's only 43. And if we divide that by the number of million years, we get a rate of loss of something like 6.3 genes per million years. There's only 43 left. And so at this rate, the Y chromosome is going to disappear in 6.8 million years. There it goes. Well, what then? Will males disappear? There they go. And if they disappear, will the human race disappear? Or maybe we could become a parthenogenic race like some lizards, a female only, and the females simply make eggs with their own genes in it. Well, that can't happen in humans or other mammals because we have a number of genes we call imprinted that only work if they come through the sperm. They're very important developmental genes. So that's not going to work. So the only chance is maybe we could evolve some new sex-determining genes if the Y chromosome disappeared. And the good news is that that's actually happened in these funny, weird rodents I told you about. The spiny rat's my favorite. There are 3 species on 3 different little islands in Japan, and one of them actually has a Y chromosome and it's got SRY on it. We don't know if it works because there's actually 100 copies of SRY and it doesn't look like it binds DNA very well. The other two species have both completely lost their Y and there is no SRY. They're both sexes have a single X chromosome, and that's a really weird story. And there must be another gene somewhere in the genome that has taken over the job of determining sex. And one suggestion is it's a gene upstream of SRY called CBX2. So we need a new sex determining gene. And the good news is it's actually quite easy to evolve new sex determining genes. You could translocate or fuse one on like the platypus AMH gene. You can have a mutant like DMRT1 mutation that made a null mutation simply sets up a dosage inequality between males and females. And that's a very common mechanism in reptiles and fish. You can have copies of that gene either that are active and form a new Y chromosome or are inhibitory. And we know examples of both of these. You can have an expression change like you do with mammal SRY, or you could even do it by epigenetic means. And we have examples of this as well. So that's no problem making new sex genes. And once you have a new sex gene is you can define a new sex chromosome. So how's that going to work? What happens if a normal XX woman mates with a spiny rat man? Well, that's not going to work very well because what you're going to get is a war of the sex genes. You might have a male determining gene and a female determining gene fighting it out in the progeny. And so what's that going to mean is there'll be a lot of intersexuality and infertility. And of course, that may well provide a species barrier and it may drive a wedge between populations of humans. So if you come back in 6.8 million years, you might find either no humans or you might find several different hominid species. And I don't know which would be the scariest outcome. So summarizing all of this, I think you can see that there's a really inexorable evolutionary forces acting against the best interests of the Y chromosome. It has degraded very rapidly. There's still 27 genes left, and the proponents of the X chromosome will tell you that, oh no, these genes can't possibly be lost, but in fact they have been lost. You don't really need any of these genes to have a fertile male. There's only 3 that are absolutely essential, but the spiny rat and the mole vole shows us that those can be lost too, and the whole Y chromosome could give way to a new sex determining system. So I hope I've convinced you that even if it's not going to happen next week, and of course it could happen next week, in fact, it could have already happened and we wouldn't be any the wiser unless we ran around the world with our test kits and made sure that every male had a Y chromosome. We wouldn't even know that the sex system had shifted and maybe there's a new Y chromosome somewhere, or maybe a new W chromosome somewhere in the human race. Thank you. A lot of interest in Neanderthal Y chromosomes and Denisovan Y chromosomes. I've never worked on those, but you can use the Y chromosome, of course, as a marker for populations of males. And that's terribly interesting because it looks like the Neanderthal Y chromosome died out and the modern Y chromosome swept in, and things like that happen a lot with the Y chromosome. So you get this hitchhiker effect if you have, for instance, a gene on a new Y chromosome which has an advantage, it will sweep over the population. And these selective sweeps, we can certainly see the imprint of them. The number of males in Europe who have Genghis Khan's Y chromosome, you would not believe. But of course, what interests me is if you go back still further and start looking at chimpanzees and gorillas. And in fact, their Y chromosomes, the chimp Y chromosome is very, very different from the human Y chromosome. It seems not to have received some of the genes from the X, and it seems to have lost a few genes as well. Oddly enough, the gorilla Y chromosome is closer to the human Y chromosome. So it looks like the chimp Y has suffered more than the human Y. And that's probably because there aren't very many chimps in the world and there are plenty of humans. Yes, yes, you're right. In fact, fish can do it as well. So it's very common to have an environmental sex determination in marine turtles, all have temperature sex determination, and so do crocodiles as well. And we're now finding that a number of reptiles can do both. So they have sex chromosomes, but if it's hot, everybody's a girl. So they're much more flexible. Well, humans are actually really rather boring because we do things in only one way, but reptiles are very interesting because there's all kinds of different X chromosomes and Y chromosomes and W chromosomes and Z chromosomes, and they flip-flop very quickly. And of course, we can't do sex, we can't do temperature sex because we're stuck at 37 degrees. And so that's probably one reason why our sex chromosomes have become so degraded. If we were frogs, for instance, we could probably flash into different kinds of sex chromosomes all the time. Sometimes sex chromosomes are only 1 or 2 million years old, and ours are 150 million years old. So they're really running out of puff. Well, people have been working on building artificial chromosomes from the point of view of adding genes for gene therapy. I haven't heard that much about it lately. It was, you know, all the rage 10 years ago or so. You can build a chromosome that has a centromere that works, it has telomeres that protect it, and you can load it up with whatever genes you like, including, I'm sure, SRY. But you know, there's better ways of getting SRY into your egg, and that's a sperm.
Jenny, thank you very much indeed. It was lovely to hear that talk. I never tire of hearing it. So listen, it gives me the greatest pleasure to introduce to you Dr. Sherman Silber. He has been working on many of the most popular and well-used fertility treatments, one for each chromosome for 46 years. He's treated many, many thousands of infertile couples who travel to St. Louis every day from all over the world. His patients come from every state in the USA, Europe, South America, the Middle East, Asia, and Africa. And among his many achievements is he performed the world's first microsurgical vasectomy reversal, as well as the first testicular transplant. I won't go through the whole of his bio at his request, except to say that I'm sure that we're all very much looking forward to his talk entitled The Y Chromosome, the difference between men and women. Sherman, over to you.
Well, thank you very much for the introduction, and thank you, Jenny, for that spectacular talk. Been following your work, oh my gosh, since the early 90s, and it's exciting. I'm going to mention a little bit about evolution of the X and the Y in terms of how it affects fertility. But I thought I'd first start out, as requested, with a little funny slide there. Wait a second. Oh, I got off that slide. Oh. I'm sorry. Well, I wanted to show that slide, but I pressed the wrong button too soon and I can't go backwards. But it was a man who was coming in from fishing, and his wife was sitting on the couch looking kind of surprised. And he came in from fishing. And, you know, we know that men and women are different, and we just don't know why. And he says to his wife, why can't you be more like a mountain stream overflowing with trout? And she's looking at him quite surprised. So that was a funny New Yorker cartoon that emphasizes that we've all been fascinated for all our lives with the difference between the sexes. And the X and the Y chromosome really has a message for that. But I'm going to emphasize the very clinical aspect. So as Jenny was saying, we all started out the X and the Y as a pair of ordinary autosomes about 300 million years ago. Oh, wait, I'm not advancing. Something's wrong. Maybe someone help. It's not advancing.
I would unshare and then share again. That'll work.
I'll share again. Screen share. I'm sorry about that. Okay, it's working now. And we just can show that over the course of time, once there was the evolution of a SRY gene, or rather a sex-determining gene, it was only of any function if it didn't recombine with its autosomal partner. And that was the origin of sex determination, chromosomal sex determination. Now, the default mechanism of sex determination really is temperature, the temperature in which the eggs are incubated. But in some animals, such as mammals, this SRY gene developed. In birds also, XX system developed, and they were able to evolve from the dinosaurs. So sex-specific, sex-determining genes develop over and over again, and they're really all different. But at least for our mammalian Y, eventually we wound up with a, as Jenny would say, a pitiful little Y chromosome, but maybe not so pitiful. The appearance of the male sex-determining gene on a non-recombining locus, that was crucial, of one of what was an X chromosome before, 300 million years ago when we were just reptiles. Subsequent evolution of mammals occurred after that, and that resulted in a generation of the developing Y because of the failure of recombination. Recombination is actually where chromosomes, routine mutations and deletions that always occur can be corrected by that process of homologous recombination. When you no longer have meiosis or recombination, the chromosomes always deteriorate and degenerate. And so that's happened in various animals, and it's resulted in the X chromosome being very well conserved, even though the Y is widely divergent. So there's really not much similarity between the Y chromosome on humans and even chimps and gorillas, let alone on mice or on Drosophila, or on worms. There's really the only similarity is the Y is short and seems to determine that it's going to be a male, and the X is bigger. And if it's XX, it's a female, and if it's XY, it's a male. But the genes are totally different. This evolved over and over and over again in different species. The ancestral Y underwent no meiotic recombination around the SRY gene. The ancestral X did undergo meiotic recombination in the female, however, and therefore the X is extremely well conserved in all mammals, whereas the Y is widely divergent. But remember that the X chromosome spends 1/3 of its evolutionary life in the testicle, where it's not recombining. That means that there are some parts of the X chromosome that are very similar to the Y. We'll get into that later, but it has to do in an important way with how the X chromosome also is very important for fertility in the male as well as the female. Now, it also relates to the non-hormonal differences between men and women. While the X chromosome remains stable, the homologous genes on its mate, really its mate was the Y, were decaying. At the same time, new genes which control spermatogenesis arrived on the decaying Y. Well, that's because transpositions occur all the time. Genes will jump from one chromosome to another all the time, and it's probably the origin of speciation, in fact. But whenever a spermatogenesis gene arrived on the Y chromosome, it stuck there because it was a safe harbor. Once on the Y, these formerly single copy genes, autosomal genes, amplified into multiple copies and achieved greater prominence. So why is that important? I'll show you later that the DAS gene, which we're all now familiar with, the most famous male infertility gene, has 4 copies on the Y. But there is one rescue copy on chromosome 3. And in fact, the DAS gene originated on chromosome 3 as what we call DASL. And when it was transposed in the monkeys to the Y, it amplified and became much more important than the original DASL. Nonetheless, in Y chromosome deleted men with AZFC deletion, you'll almost always find some sperm because chromosome 3 has a DAS homolog. So we have delay of ancestral autosomal genes on the heterogametic sex chromosome and parallel accumulation of genes that controls spermatogenesis. Because as Jenny said, I mean, the Y would have completely disappeared, except that we have this accumulation of genes from all over the genome that controls spermatogenesis. Now, X inactivation is a very important subject because when these genes decay on the Y, so you would think that women have 2 copies on, because they have 2 X chromosomes of these intelligence genes and other genes, and men only have 1 copy that's functional. You would imagine that, women would be twice as intelligent as men. Now, I know they're more intelligent, but they're not twice as intelligent. So, in fact, men would be dead if it weren't for some mechanism to give us equal expression of X genes. So over time, the ancestral Y shrunk because of decay of X homologous genes. But the loss of those ancestral Y genes were compensated for by hyperexpression of Y homologs on the X and X inactivation. In other words, those genes on the X that had decayed on the Y become hyperexpressed, so that in the male, at least, he has as much expression of those X genes as the female. On the other hand, you'd have too much X expression, and the female couldn't survive if there wasn't X inactivation, where half of the cells in the body actually have the X chromosome inactivated, so there's no expression. There are genes that escape X inactivation, and we're going to point out later that that is probably one of the most important reasons why there are non-hormonal differences between men and women. For non-hormonal differences being like autism, more common in males, or like autoimmune disease, more common in females. So this way, males did not suffer from a loss of genes, but females did not have twice as many functioning X genes as the males, and so we were all relatively equal. And this is just a diagram from Jacalian and Page paper on this in 1998, showing this is a gradual process. First, we developed a pseudo-autosomal gene, and the X-linked gene was not X inactivated, and the Y-linked gene had a reduced expression, and the X had increased expression, and then eventually you had to have X inactivation so that both the male and the female would have equivalent expression of what were extremely important X genes, which were really autosomal genes. So we know that for years, we just looked at the karyotype and we thought the Y chromosome had nothing to do with anything except sex determination. But then when we did our first study in 1995, just mapping the Y chromosome, and it was the first mapping of any chromosome, we only picked the Y because it was short, and we thought that it might have something to do with spermatogenesis. As it turns out, it's probably the most difficult chromosome in the whole genome to sequence because of multiple repeat copies. So anyway, so there were no deletions detected in 90 fertile males that were azospermic. But we detected these deletions in what we call the AZFC region in 13% of azospermic males. And that was the origin of the AZFC deletion. However, then we got in much more detail, because this mapping program was maybe 1 every 30,000 base pairs. So, I mean, you're like trying to find a needle in a haystack. And when we looked at it in more detail with AZFA, AZFB, and AZFC regions, still not enough detail, we were able to find that the common deletion was in this AZFC region, which resulted in a combination of maturation arrest and Sertoli cell only. There were often some sperm located. And when we did more detailed test studies, what we found was in this, if you had an AZFC deletion, there were almost always some sperm, either in the ejaculate with an extremely low count, or in the testes if you did a difficult microsurgical testa procedure. And where did all this happen? Well, the original insight to hybridization studies showed the JAS transcripts in the spermatogonia, which explains why DAS is probably the most important original spermatogenesis or even oogenesis gene, but also why you could have a combination of maturationist and Sertoli cell only, because it's expressed in spermatogonia. Now, as I said before, there's no relationship to any of the Y chromosomes in Drosophila or worms or even fish or even mammals. They only are called Y because they're smaller than their sex chromosomal counterpart, but they're all different. And so DAS is found in the Y chromosome only in humans and in higher primates, because there was a transposition from chromosome 3 to the Y between the transition between old world monkeys and new world monkeys. And dazzle is found on chromosome 17 of the mouse, but not on the Y. And the mouse has a gigantic Y chromosome. Dazzle homolog found on chromosome 3 of humans, 1 copy, but DAS is found in multiple copies in the Y. So the DAS homolog, it's just like DAS, the Boole gene it's called, is found on an autosome of Drosophila, but not on the Drosophila's Y. So it's all very confusing, except that evolution finds a way over and over again of promoting a genetic sex determining system rather than a temperature dependent sex determining system. But also from a clinical point of view, this shows why in men with AZFC deletion, that's the very best prognosis for azoospermic men. And when we sequenced the whole AZFC region, we understood what the problem was. In this region we call B1 and B2, there were 229,000 identical base pairs. And within between them were 19 transcription units, 7 families of genes, including DAS. And there was a whole palindromic complex that actually exceeded that B2B2 pair, in which there were 27 transcription units, 11 families of genes that included RBMY. So we finally found out the reason why these DAS deletion, why these AZFC deletions occur, because illegitimate homologous combination, I'll discuss in a section, is where these B2, B1 genes recombine with each other and the middle drops out. So what's the mechanism of de novo appearance of these deletions? It's an inherent result of the structure of the Y. In other words, the Y chromosome has absorbed all these genes and amplified them that helps increase spermatogenesis, but then spermatogenesis is dependent on the Y. And the Y, as Jenny said, is very fragile. So what about the sequence of AZFC? We call it illegitimate homologous recombination. If you look at them, you can see that these recombination targets are identical, and what's in between them will disappear when they recombine. Now, all chromosomes must recombine to maintain themselves over time. This is a little bit of a funny slide. Autosomes repair themselves by recombining with each other during meiosis. Okay, everybody knows that. The Y tries to repair itself by recombining with itself. The DNA between identical sequence of nucleotides drops out when homologous sequences on the same chromosome recombine. And this is just a diagram of how the DAS gene was transposed from chromosome 3 in humans to the Y in humans. And this is just showing that chromosome 3 has a DAS-like or DAS-old gene, which is a DAS homolog that rescues spermatogenesis in non-obstructive azoospermia. Now, what about the offspring? Are the offspring of men who have ICSI that have a Y deletion, are they going to have an amplified Y deletion? Is it going to get worse? Or will they have no Y deletion? Well, it's obvious that they have exactly the same Y deletion as their father. But where did it all come from? The father of AZFC deleted men, they're not Y deleted and they're obviously fertile, but the son is Y deleted. Another example, father of AZFC deleted man is not Y deleted. And he obviously had to have good spermatogenesis because he had all these children, including a son. And the son was AZFC Y deleted. One boy from Tessa ICSI blood was AZFC Y deleted. So Y deletion occurs de novo in the fertile father's testes. And clearly the father and brother here were obviously fertile, but this Y deleted occurrence was something that happened within accidentally. The Y deleted sperm from his father, and it's probably 1 in 2,000 sperm, and every man with normal spermatogenesis has a Y deletion or Y dropout. If that's the lucky sperm that fertilizes mom's eggs, then the baby will be Y deleted and will be infertile. And so there's no transmission of the Y deletion, no increase in transmission of a more expanded deletion in the son of AZFC mutant men, but they will have the same AZFC deletion as their father. So why does the Y not completely disappear? Well, because autosomes repair themselves by recombining with each other during meiosis. The Y tries to repair itself by recombining with itself. And this is the funny part of it. Autosomes have sex with each other. The Y masturbates. And that's my humorous way of trying to think about it. And this is a picture from the Howard Hughes Institute, where we see that what happens is because you have so many identical repeats, the Y tries to repair itself. It's not that efficient, but it's not as efficient as meiosis, but it tries to repair an area of the Y that was injured by mutation just by gene transfer. And that's one reason why the Y has held up over all these years. And it may be an argument against Jenny's postulate that the Y will eventually disappear. So this also explains multiple copies of Y genes. The Y is so difficult to sequence because of these multiple arrays of repeat sequence, which we call amplicons. Once DAS transposed from chromosome 3 to the Y, it amplified into multiple copies, but the original homolog of DAS remains on chromosome 3 as a backup. And now the complete sequence of the Y chromosome was almost impossible to do because there are so many of these ampliconic repeats, and really nobody else has even done it. And we need to be able to have other people sequence amplicons, because I think there's tremendous dark DNA in these amplicons that'll explain a lot of male-female differences. So we had to have, with these huge palindromes of mirror image identical infertility repeats, we had to look for minute variants and otherwise identical repeats used as sequence family variants, and we had to do it in just one man's Y chromosome. So what we found was not only are there these multiple amplificated repeats, but there's also palindromes. What is a palindrome? Palindrome is where one side or the other side of the center point is identical in reverse order. And this is a perfect example of a palindrome. Madam, I am Adam. If you look at the center, you'll see that the either side is actually identical, but in reverse order. Madam, I am Madam. And so that's what is composed of most of this ampliconic region, are not only sequence repeats, but sequence inversions as well. And those sequence inversions are another way that we can just lose huge areas of the Y chromosome. And that's how we lost AZFB. AZFB deletions take out a gigantic section, not just 229,000 base pairs, but a total of 4.9 million base pairs. So what about testis-specific versus ubiquitous expression that Jenny was talking about? So there's three different areas of the Y chromosome. There, after we sequence the whole thing, there's the X transposed, which is identical in the female and the male. And that they have to avoid X inactivation or we wouldn't be alive. There's the ampliconic region, which is huge, expressed only in the testes. And then there's the ubiquitously expressed X degenerate region, 16 genes that appear to be degenerated from the X homologs. They're different, but they're different. And that means that the X on homolog is most likely hyperexpressed. So this X degenerate region, which escapes X inactivation, is probably one of the most important areas to study to try to understand why men and women are different if we remove the hormonal differences. Smaller Y deletions encompassing fewer spermatogenesis genes, by the way, like GRGR, causes varying degrees of oligospermia instead of azoospermia. So we can summarize what the Y chromosome looks like in a simple graph with AZFB and AZFA and AZFC lesions. And we prefer to be more specific based on sequence, like P1, P5, and P4, P1, and B2, B4, GRGR. And the reason for that is take a look at these GRGR deletions. That's like half of the AZFC deletion is deleted. And that means that these men will have lower sperm counts. It's even a polymorphism, the GRG lesion, for a greater risk of male infertility. So why don't human males have really low sperm counts? Well, it's sperm competition. And this is what the famous Roger Short, who is my idol, figured out, that chimpanzees have relations with each other as soon as one female goes into heat. Every male in the pack will mate with that female. So it's likely that the male will have the sperm count of the male offspring will have the sperm count of the male that had the highest sperm count. Now, so you see that chimpanzees have gigantic testicles and just humongous sperm counts. What about the gorilla? This fantastically macho animal. There's no sperm competition. And he has maybe two or three females, and they never have a chance to mate with any other males. And look at their tiny little testicles. I mean, the gorillas really have tiny testicles because of the lack of sperm competition. And the Y chromosome in the chimpanzees is enormous. They have much more amplicons than the humans have, and the gorillas have less amplicons. So small deletions cause less severe spermatogenic defects. AZFC deletions remove 3.5 million base pairs. Sorry, I didn't mean thousand, I meant million. And AZFB deletions remove 6 and 8 million base pairs between 32 and 42 genes instead of 21 genes. So AZFB deletions so far seems to result in no spermatogenesis except maturation arrest. Small deletions like GR, however, remove only 9 genes, and they are only basically polymorphisms for lower sperm count. And this is, you see, GR deletions occur between identical regions within the AZFC that take out only a smaller amount of AZFC and leave half of the DAS genes intact. So our conclusions are milder degrees of oligospermia are caused by smaller deletions, which take out only some copies of male-specific genes, leaving intact back copies. And sperm can be found in tiny numbers in Tessa cases if there is just one backup copy of a deleted gene. So AZFC is an example of a very good prognosis for Tessa case, as well as GR deletions. Sperm count varies with gene copy number, and testis-specific genes on the Y always have multiple near identical copies. Small deletions are more common than larger deletions, and that makes sense. And these more common deletions leave backup spermatogenesis genes that result in lesser degrees of spermatogenic defect. So we can look at a graph of a bell-shaped curve of a normal population with sperm count and of a population that's AZFC deleted. And they're going to have sperm. And then the GR deleted group falls in between AZF deleted and a normal population. So we think that this is going to be the way we have to look for the genetics of azospermia and oligospermia throughout the rest of the genome, but we've been stymied in the Y chromosome to find anything more. Turner syndrome is fascinating. I won't go into it in detail, but what if the whole chromosome turned into a palindrome? Well, then what you wind up having is XO is not always the absence of an X chromosome in a baby girl, but rather the absence of a Y chromosome in a baby boy. There's no maternal age effect in XO Turner syndrome. And in 3/4 of XO girls, the X chromosome is of maternal origin. So in the majority of Turner syndrome, we would guess we're not going to find any sperm because of the majority of them, and we're not going to find eggs because the majority of them are males that lack a Y chromosome rather than females that lack an X chromosome. And what about the X chromosome in general? Like the Y, the X is present as a single copy in the male. Therefore, mutations in the X that are favorable to spermatogenesis will be readily expressed in male offspring. So the X spends 1/3 of its evolutionary life in the testes where it cannot recombine, and the human genome has not been completely sequenced. We know that. And so because of all these amplicons, we do not have complete quality sequence. So I call that the dark side of the X. The X cannot escape completely some of the problems the Y solves by constructing amplicons and palindromes. In fact, 12% of the X only undergoes meiosis the X only undergoes meiosis in the ovary, not in the testes, where it spends 1/3 of its life. So it therefore has huge ampliconic regions. 12% of the X is just like the Y. And these regions are still completely unsequenced, very likely to have a rich complement of genes for both spermatogenesis and oogenesis. But one other thing, what about the difference between the sexes? The X degenerate portion of the Y is going to be different in men than in women, especially if they escape X inactivation and hyperexpression. Autism is more common in males. Could that be from the dark side of the X? Well, actually, now we have evidence it may be really related to the Y chromosome X degenerate genes. And autoimmune disease is more common in females. Well, all this could be related to the fact that there are differences between the sexes that are related to the X and the Y chromosome, but are not hormonal. So I'm not going to go any further. Our time is up, but I can answer any questions about what I think is extinction in general and how animals go extinct and why the dinosaurs went extinct. It's one of my favorite subjects. But my time is up now, so I'll close now and I'll give it back to the, I have other slides, but I think my time is up, so I'll stop with that.
Yeah, thank you, Professor Sherman. That was brilliant and very comprehensive. So I'll take you up on your most favorite topic. I read earlier as well that you've always been fascinated with the dinosaurs as a kid and even as an adult. So could you please elaborate a little more on that?
Yeah, I love this subject because extinction is always interesting and confusing because none of us really would like to go extinct, but we will, of course. And the default determination of the sex of any offspring is really temperature in which the eggs are incubated. And we see in about 1/3 of fish, the determination of sex is just temperature. We see in reptiles, almost all the reptiles, the determination is temperature. And yet in birds, it's an XXXY sex determining, genetic determining system. In mammals, it's obviously an SRY related Y chromosomal sex determining system. In Drosophila, Y chromosome is sex determining, even though it has nothing to do with DAS. And in fact, DAS is actually a gene that is important for both spermatogenesis and oogenesis. It's a very primitive gene. And so why would we have the evolution of these these genetic sex determining systems we call GSD? Why would we have over and over again different evolution of different Y chromosomes, of different XX systems, of different Z chromosomes? They're all different from each other, except for the fact that one is small and one is big. And why does that happen over and over again? There has to be some evolutionary benefit to that. And the first thing that comes to mind is that if you have a temperature dependent system, then whether the male-female ratio is going to be purely dependent upon the ambient temperature. And the evolution of a genetic sex determining system, a GSD, will assure a 50-50 or at least a relatively equivalent gender ratio for reproduction. Now we know from Antarctic ice cores that every 100,000 years, for at least the last 800,000 years we have data for, and I think it's now going back to over 1 million years, that every 100,000 years, for whatever reasons we don't understand, long before humans, the temperature gradually goes down over 100,000 years actually in stages of 25,000 years each. And then after 100,000 years, there's a sudden spike and the temperature goes right back up again to what it was 100,000 years ago. And then it gradually goes down again. And then it keeps on happening every 100,000 years. That is in relation to the humans. Now, so we understand the current global warming situation. When we look at CO2 in the Antarctic ice cores, suddenly now, since industrial humans, it's just going way higher than it ever went before. So it's different now than it was in the previous 800,000 years, but nonetheless, temperature change is obvious. We've had great ice ages and the dinosaurs clearly lived in a very warm climate. And so when the asteroid hit 65 million years ago, which everybody knows about, there had to be a tremendous change in the environment. But it couldn't be just toxicity because mammals survived and birds survived, and even crocodiles survived. So why is that? The temperature change, and our math tells us that most probably it was temperature going down, which resulted in 90% males. And in 3 generations, 90% males would have resulted in basically the extinction of the dinosaurs. Could have been 100% males, but even 90% males mathematically would result in dinosaur extinction within 3 generations. And why did the crocodiles survive? Well, they have a whole different system of laying eggs. They go inland and, sorry, they don't go inland like dinosaurs. Nests are always found in inland areas, but they go up and down estuaries and streams. And so they can lay their eggs in their nests, crocodilians can, and vary the temperature. And the outer eggs, of course, are colder and the inner eggs are warmer. And so that's how they have not exactly an equal sex ratio, but they have enough of a variation in sex that they were able to survive, as did turtles for the same reason. But otherwise, GSD animals really didn't survive, and the animal of sex determination survived 65 million years ago.
Well, Sharon, I'm going to challenge you on that a little bit, because some people would say the dinosaurs absolutely did survive. They're flying around with feathers right now. So let's continue this debate at some
well,
other
100%.
time.
Birds are dinosaurs that evolved a ZZZW system, so they survived.
But not until the Neognaths diverged, only 100. Yeah, so let's continue this at some other time. Listen, I'm going to bring in Jenny Graves here. Jenny, we've listened to your fantastic talk here, and it's lovely to hear from you. I can see that you're online, and we do have a question for you. It's from Jacques, who says it's a great and fascinating talk. But to summarize his question, we're finding out a little bit more about the Neanderthal and Denisovan Y chromosomes here, and he suggests that these studies have shown that the Homo sapiens Y chromosome completely replaced them. So although it's small, isn't it powerful from an evolutionary point of view? So do we need to expand this work, not looking back 10 million years, but perhaps looking back to 200,000 years? Jenny, what's your opinion?
Well, Y chromosomes, because of their nature, do turn over very rapidly. And there's one phenomenon that I didn't mention, I don't think Sherman did either, and that's called genetic hitchhiking. So if a particularly good variant turns up on a Y chromosome, makes more sperm or longer sperm or faster sperm, that variant Y will carry all before it, no matter what the other genes on the Y chromosome are. And that's probably why the Y chromosome has been reduced, and very rapidly, because there have been what are called selective sweeps. And this may very well have been what occurred to wipe out the Neanderthal Y. If an invading Y chromosome has a variant that makes more sperm or better sperm or faster sperm, it will very rapidly replace other Y chromosomes. And of course, it will carry with it, because there's no recombination of the Y chromosome, that variant Y will carry other variants as well, some good, some bad, some indifferent. So I don't think there's anything funny about one Y being rapidly replaced by another Y. And that can happen, and it's probably happening all over the place now. It probably was much more obvious when the population was smaller, because then selective sweeps could be complete and not just local like they are now.
Question from Denny Sarkas, Jenny. What's the evolutionary advantage of losing the Y?
Well, the funny thing about the Y is that evolution doesn't work very well. You'd think the evolution would save the Y, make the Y better, stop it being reduced. But in fact, that's not what happens. And as Sherman said over and over again, you're getting loss of active genes from the Y. And I think you can't say that loss is an evolutionary advantage, but it's inevitable because of the nature of the Y chromosome. So it's a lovely example of what I call dumb design, which of course is the reverse of intelligent design. And a lot of things in life that you see are, I think, evolutionary accidents that may not be good in themselves, but once they've happened, they can't unhappen.
There's Carlos Hernandez asking Dr. Sherman, could CRISPR-Cas9 have any utility in repairing the AZF-C deletions?
This is something we're really talking about because we've got a project now with Kyle Orwig and Amanda Clark and Katsuhiko Hayashi of taking skin biopsies from azoospermic men and converting them into iPS cells and the iPS cells into PGCs, and eventually it'll be into SSCs and into sperm. But if we identify that the problem is a specific deletion, then it isn't really popular right now, the idea of using CRISPR for germ cells, but you could use CRISPR for the stem cells, and that would be allowed. And then you convert those stem cells into PGCs that actually have repaired AZFC, and then at least then you would have spermatogenic stem cells that were normal. So, I mean, it's not immediate, but I think that's going to be the future, is the plasticity of stem cells and using CRISPR in the stem cells and then creating germ cells from those stem cells.
Okay, shall I go with the next one, Stacy, or would you like to?
Yeah, I have a question for
Go for it.
Professor Graves. Thank you for your wonderful lecture. I had actually met you when you came over to India a couple of years ago, and we discussed a lot about the role of the Y chromosome, the emerging roles of the Y chromosome. My question to you, Dr. Graves, is there are new studies reporting new roles of the Y chromosome genes, going as far as autoimmune and neuropsychiatry roles, and as well as a post that you wrote somewhere about the sex bias in COVID-related deaths. So how do you reconcile these new functions attributed to the Y chromosome genes with the standpoint you've always held on to regarding the Y chromosome degeneration?
Well, it's much worse than that, Stacy. People have looked at expression of all the genes in the human, in males and females. And the shocking thing is that 1/3 of our genes are expressed differently in males and females.
Right.
So how does the Y chromosome accomplish that? Presumably, a lot of these are hormone regulated genes. So the role of the Y chromosome in directing a testis formation is indirectly causes a lot of these differences in gene expression. And these genes are not just in the gonads, they're all over the body. So a gene in the liver, which is not on the Y chromosome or on the X chromosome, will be differently expressed in males and females. Well, why? What is it about the Y chromosome? And I would agree with Sherman that it's not all about the SRY gene. There are another 17 or so genes in the area which are differentially expressed. So some of these genes, the dosage may well have an effect on other genes in the cascade. So there's a lot we don't know, but we now have a very complete readout of the active genes on the Y chromosome. So we can look at those activities in different tissues, and we will find that they're not all about sex. So I wouldn't call it dark matter anymore, because I think we actually know what genes are there, but we really don't know what they do, and we really don't know what tissue. And we're still getting surprises. I mean, we know now that SRY itself is expressed in the brain. Now, is it having an effect on the thought of men? We don't know about that. It may have an effect on the propensity of men to get Parkinson's disease, for instance. So we can't just say, oh, well, there's only 27 genes on the Y. We know all about them. We don't know all about them. And particularly, we don't know about their knock-on effects, of which there are lots. I mean, one thing I should say is a lot of these genes in the X degenerate part of the Y chromosome are not inactivated on the X. So it looks like the reason they're still on the Y is actually to balance the dosage of genes in males and females. So there's 2 X copies in a female, and there's an X and an active Y copy in the male. And so that may be why they're still there. It's really hard to lose them if it has terrible effects on processes. So it's not all about sex.
So thank you, Jenny. Jenny, so I've got 2 questions, similar ones, one from Murad from Canada and one from Hassam from near you in Melbourne. They're both asking about SRY relocating itself onto another chromosome. So Murad is asking, can it? And then Hassam is referring to the, it does occur in XX males, for instance. So how is it that it's sometimes just the SRY and not necessarily the rest of the chromosome? So can you just comment on that in generally and maybe address those specific points?
Well, I think Sherman covered some of these, but there are patches of the Y chromosome which are homologous to patches on the X chromosome. And although they don't normally pair at meiosis, they can recombine. And that's why you can get the tip of the Y chromosome swapping places with the tip of the X chromosome. So not just in the pseudo-autosomal region, which is shared, but further down. And if that happens, you'll transfer the SRY gene to the tip of the X. And so you get a phenotypic male. But of course, you haven't transferred all the fertility genes in the azoospermia regions on the long arm. So what you have is an infertile male. So those would be the XX males who are actually not that rare.
I have a funny story about that I can tell you quickly. It was at a ASRM meeting, and Peter Brisden, who's a brilliant embryologist and infertility doctor, was talking about the psychology of an XX male. And he said to a big audience, I don't know how you counsel these men to tell them they're really a woman with 2 X chromosomes. And I got up there to answer, and I said, well, just tell him that he's got twice as many intelligence genes as most men. He shouldn't be unhappy about being XX.
There is one question for you, Dr. Sherman. Somebody wants to know the link between autism and Y chromosome microdeletions.
Okay, well, let me, there are 2 possibilities. One is we know that we don't know what's going on in the apliconic region of the X. And, you know, I mean, it's obvious from X-linked recessive diseases that are plentiful, hemophilia being the common one everybody thinks about, and Duchenne's muscular dystrophy, another common one. I mean, it would just make sense that if a disease or a problem is 4 or 5 times more common in males, it could be transferred from mother to son from the X chromosome, and you know, half of the children would have the problem and half wouldn't. So that's a possibility. It has to be discovered. But also, I'll tell you something fascinating. We haven't published this at all, but the whole purpose of this is to turn people on, and people that are really actively doing this sequencing might want to look at this. And we were happy to send the material to everyone. So we have a patient who had IVF patient who had identical twin or homozygous twins that were both autism. And she joined a chat group, and a lot of chat groups like this. So that brings these people together. And she is in a chat group where all the women had donor sperm. She was a single woman, had donor sperm from the same particular donor and egg bank, over 50 cases, which should never happen. I mean, the sperm bank should never be doing that, but they did. And all these women, their female offspring are just fine, but the males all have some degree of autism spectrum. So that makes you really think, could it even be on these genes on the Y chromosome? And, you know, I just say that openly here. We haven't published anything, but it's available. We can send this material to these patients that agree to do it to anyone who wants to work on that.
Jenny, one from Michael Reed. Was sex reversal in avian species that can occur due to damage or removal of the functional gonad? Okay, so in these birds, do you see any changes in the sex chromosome from the vestigial gonad? So anything in structure, methylation, or something like that?
Not as far as I know. I mean, birds are terribly interesting because, of course, the other way round, it's the male that is a homogametic sex with two Z chromosomes, and the female has a Z and a W chromosome. But birds are also strange because only one gonad develops as a testis, and if that testis is removed, the other gonad will develop into an ovary, and sometimes you'll get sex change, but you won't actually have differences in sex chromosomes. But we still don't really understand how the Z and the W chromosomes determine sex. We know there's a gene on the Z chromosome which is lacking from the W. It's a gene called DMRT1, which has been associated with sex for probably 1 billion years. It's even associated with sex in Drosophila. But that's not the whole story. So it seems to be dosage of that gene, 2 copies and you're a male, a single copy and you're a female. But that's not the whole story, because you can get birds that are hermaphrodites. One half is female and has a single Z chromosome, and the other half is male and has 2 Z chromosomes. And you think, well, how can that happen? Because DMRT1 would not be cell specific, and nor would all the downstream actions, and certainly not hormones. So there must be something else. And we still really don't understand what that something else is. Presumably it's something else either on the Z or the W chromosome, but nobody's been able to demonstrate any female specific genes on the W chromosome. But there must be something there, and we don't know what it is.
You know, it's so confusing in that like the SRY gene is just a trigger. It doesn't know how to make a testicle. I don't know, Jenny, how many genes, there must be so many genes that contribute to make a testicle that we don't know anything about.
No, exactly. I think we have about 60 genes in the so-called pathway to gonad differentiation, but I bet there's a lot more than that. And we keep on discovering patients who have a mutation in a different gene, and then it turns out that that gene has a knock-on effect on another gene that has a knock-on effect or inhibits SRY or SOX9 or the downstream genes. So it's not a pathway. It's more like a network, and it's sort of full of push me, pull yous, where you have inhibitors of inhibitors and activators of activators. And again, it's a wonderful example of dumb design, because if you or I were going to design a sex determining system, we do A goes to B goes to C goes to testis. And E goes to F, goes to G, goes to ovary, wouldn't we? We wouldn't be doing any of this A's and B's and C's and D's getting all mixed up. It's crazy.
Okay, folks, we are going to wrap up there. This has been absolutely fascinating. Thank you very much to everyone who took part. I'm told I've got to thank myself. So thank you, Darren. There we go. Thank you very much to Stacy, to Sherman, of course, and for Jenny for getting up so early. Thank you very much to our organizing committee of this initiative, I3, who produced these webinars. Jacques Cohen, who asked a question, Peter Naj, Shaista Sanruddin, Thomas Elliott, and Marianne Saviches, and finally, the inimitable Giles Palmer.
We look forward to catching up with you all again, and our next session is on the 30th of November, the Ministry of Embryology and Implantation, looking at mitotic divisions, embryo development and selection, with talks by Daniel Linka, with Saspa Pribensky, Mina Alikani, Lorena Bori, Thorir Harjison, Dean Morbeck, and more. Thank you and see you soon.
Thank you very much, everyone. Cheers. Bye now.